Hi everyone, here we are in microbiology. We're on our third video on prokaryotic cells and we're in section 3.3. And the first learning outcome we have is name and recognize the two parts of the cell envelope and state their major functions. So far we're kind of working from the outside of the bacteria and coming in. So we talked about flagella, we talked about glycocalyces like capsules, and now we're going to the next layer. If you look at this picture right here, they're showing you right here is the cell wall. So if you're going from the outside, you would have your glycocalyx, your slime layer or your capsule. You'd get a little deeper and you'd get to the cell wall. And inside that's going to be the plasma membrane. So we called this, all this together actually is what we would call an envelope. But the two main parts that all cells would have, or almost all cells would have, that would make a cell envelope would be the cell wall and the plasma membrane. I think your book is using the term cytoplasmic membrane, and plasma membrane, cytoplasmic membrane, and cell membrane all mean the same thing. Okay, so when we're talking about the envelope, the main parts of the envelope, we're talking about the cell wall and the plasma membrane, or the cytoplasmic membrane. Oh, I didn't mean to put that right there. Okay, so let's talk about this cell wall. We're going to look at the chemical structure of that. And what I have on here, it says differentiate between the structure of the gram-positive and gram-negative cell wall. And gram-positive and gram-negative is a really important concept in microbiology and in healthcare. So I'm going to try to convince you that you should know a little bit about this and have a little understanding. But turns out if you look at bacterial cells, this cell wall that kind of looks pretty boring in that diagram, there's actually two major types. And some cell walls are gram-positive and some are gram-negative. And this was figured out by a guy with the last name of Graham in the late 1800s, that he was just trying to find ways to figure out, how do we identify bacteria? Will some bacteria stain differently? And this scientist found that some bacteria stain purple, and he called them gram-positive, and I'll repeat that a bunch of times. And some bacteria using a certain technique stained red, and he called those gram-negative. So first thing we want to look at is, here's that plasma membrane. So if you look at this diagram, I put this same diagram in your study guide because just in case you wanted to write on it. But inside here, this would be the inside of a cell. So this is the plasma membrane, and then here's the cell wall right here. Okay, here's the plasma membrane, and there's the cell wall. So if I go back here, there's that plasma membrane and the cell wall. Okay. So in gram-positive cells, they have this weird protein carbohydrate thing called peptidoglycan. And that sounds like a really weird word, but it's a word that you'll get used to. You might have noticed that I was just kind of hesitating when I was speaking in the past sentence or two, and it was because I was noticing that right here they spelled it peptidoglycan. They spelled it wrong, and then all of a sudden I went, have I been spelling it wrong all my life? And no, I'm spelling it right. This is how you spell it, peptidoglycan. Not a big deal. I just caught my eye and threw me off. Anyway, gram-positive cells. So you have the inside of the cell, you have the cytoplasmic membrane, and then outside of that, in gram-positive cells, you have this thick layer of peptidoglycan, which is a mixture of proteins and different kinds of carbohydrates. In gram-negative cells, you have the same cytoplasmic membrane, but you have a thin layer of this stuff called peptidoglycan. And then on the outside of the cell, outside of that wall, this is still part of the wall, there's another membrane. You have an outer membrane. And in gram-negative walls, in that outer membrane, we have something called lipopolysaccharides that you can just call LPS. And those are actually kind of important in causing disease. So Later in the semester, we're going to talk about how bacteria can produce toxins and make us sick by these toxins. And this lipopolysaccharide in gram-negative cells does act as a toxin. So we'll be talking about that. So I want you to remember that. So what I want you to remember about the difference in structure of gram-positive and gram-negative, I always do this. I draw myself a simple line drawing, like in a live class. I would have students do this. I would say, here's the inside of the cell. There's your plasma membrane. And then gram-positive cells have a thick layer of peptidoglycan. Gram-negative cells also have a plasma membrane, but they have a thin layer of peptidoglycan. They have this outer membrane that has this stuff called lipopolysaccharide, this LPS. Okay, so that's what I want you to know about the difference of gram-positive and gram-negative walls as far as structure. Now, we do something in the lab. Hospital labs do this all the time. And microbiology students do it a lot. You would get a couple chances to do it. You guys are going to do it using virtual labs. But you're going to do a gram stain. And so it says, explain how the cell wall structure determines how the gram stain works. So you're going to go over this in lab, but I'm also going to talk about it here. So what you want to do is you want to know, are your cells gram positive or are they gram negative? And I'll explain why you would want to know that in a couple minutes. But here I have a slide and you put some bacteria on it. And you take a stain called crystal violet. And crystal violet is really dark purpley blue. And you put that on your slide, and no matter what cells you have, whether they're gram positive or gram negative, they're all going to stain purple. And if I look back at my picture here, what happens is the crystal violet kind of goes through this wall. The peptidoglycan, it gives the wall a lot of structure. It makes it kind of hard and rigid, but it's more like a basket. So it's like having a basket. So it's got kind of spaces where crystal violet can kind of squeeze in and land in here. And the same thing for this, that crystal violet kind of gets here and sits in there. So both gram positive and gram negative walls will stain purple. The second step of the gram stain is you add some iodine. And iodine is something we call mordant. And the mordant kind of helps the stain attach to the cell. Well, it turns out here we have our crystal violet goes in, and the iodine goes in too. And it attaches to the crystal violet. And it makes it bigger. Okay, so crystal violet iodine is bigger than crystal violet or iodine alone. And the same thing here. The crystal violet and the iodine attach to each other. So if you look at your cells under the microscope, the iodine doesn't give it much color. They're still going to look purple from the crystal violet. The third step, you're going to add some alcohol. And crystal violet and iodine will rinse out. It will decolor. It's a decolorizer. It'll rinse out the color. Except in this gram-positive wall, this layer is so thick that big crystal violet iodine, remember the crystal violet went in, the iodine went in, but when they go out, they're stuck together and they're too big to get out. This multi-layer thick peptidoglycan prevents them from getting out, even if you add alcohol. This thin little layer of peptidoglycan, the crystal violet and the iodine can get through that. So the alcohol will rinse it out. So in the gram-positive cells with a thick peptidoglycan, they remain purple after you rinse it with alcohol. Whereas the gram-negative cells with that thin layer of peptidoglycan, the crystal violet and iodine just rinses out. So now they're colorless. They don't have any color. And it's hard to look at cells under the microscope that don't have color. So we add a counter stain or a secondary stain, and it's a red stain called safranin. And the red doesn't show up very much on these purple cells. The purple's kind of more intense. But it will stain the gram-negatives from that colorless to red. So at the end of the gram stain, Gram-positive stains stain purple, and I'm emphasizing my P's to help you remember that positive and purple go together. Gram-negative cells stain red. Now, the reality is they kind of look pink. Like that almost looks a little bit pink, doesn't it? But I want to say I don't want to say gram-negative cells turn pink because that doesn't give me positive purple. It mixes my P's up. So I always say gram-positive cells turn purple, gram-negative cells are red after a gram stain. Gram stain is a really common, really useful technique in the lab. So here's some cells after a gram stain. Here's some purple ones, here's some red ones. And so these would be gram-positive and these are gram-negative. And it's actually quite easy to tell. The stain is really fast to do, and even a beginning student can tell them apart. So again, here's a mixed culture. There's more than one kind of bacteria, but I can see that there's some gram-positive Staphylococcus, and there's some gram-negative Bacillus. Turns out the kind of bacteria that causes staph infections is a gram-positive Staphylococcus. So that's what a staph infection bacteria would look like. Things like E. coli are gram-negative Bacillus. So why do we care? What's the clinical importance of the gram stain? And one of the big things is it helps us identify them. And that sounds like, gosh, if there's like thousands of different kinds of bacteria, and you can only put them into 2 categories, why is that a big deal? But it is a big deal. Think about if, someone robbed a store and you said, I don't know anything about who robbed the store except I noticed that it was a woman. You just eliminated all men. So you just by having, this or that, gram-positive or gram-negative, you can eliminate half the bacteria. So that is a big deal. And another reason why this is clinically important is gram-positive cells are actually more sensitive to antibiotics like penicillin. Turns out penicillin attacks peptidoglycan. And so if your bacteria are growing in the presence of penicillin, the antibiotic penicillin, you've taken some medicine, the bacteria fall apart. That wall is no longer rigid and the bacteria don't grow normally and they die. Because the gram-negative cells have this outer membrane, they protect their peptidoglycan. So gram-negative cells aren't very sensitive to penicillin. And so you wouldn't treat an E. coli infection. E. coli is gram-negative. You wouldn't treat an E. coli infection with penicillin. So it's something that you can do is if you do a gram stain, you might not have identified the bacteria, but you know that, oh, this bacteria might be killed with penicillin. This one, I wouldn't even try. Okay, so if I put my P's together, I can say gram-positive has a thick peptidoglycan, stains purple with the gram stain, and is penicillin sensitive. Okay, gram-positives have a thick peptidoglycan, penicillin sensitive, stains purple in a gram stain. Gram-negative have a thin peptidoglycan. We don't have all the P's together with gram-negative. So gram-negative has a thin peptidoglycan, stains red with the gram stain, and is more penicillin resistant, just naturally resistant. Okay. Cell walls. Oh, here's a gram stain again. It says, tell me the shape and the arrangement in the gram reaction. So these cells kind of look, see if you can say they're gram-positive or negative. Gram-positive. The shape is coccus. They're round. The arrangement, they're in clusters, so it's called Staphylococcus. So this is gram-positive Staphylococcus. Here's one. Oh, it's red. So that makes it gram-negative. They're elongated, so they're bacillus. They don't have a particular arrangement. They're just singles. So we've got gram-negative bacillus, and it turns out that's Staphylococcus. This is Escherichia, as in Staph aureus and E. coli. The last thing in section 3.3, it asks you, we were talking about the envelope. Remember, the envelope has the cell wall and that plasma membrane or the cytoplasmic membrane. probably remember something about plasma membranes from another biology class, that they are made of that phospholipid bilayer with proteins embedded in them. Remember, the major function of the cytoplasmic membrane is to control what goes in and out of the cell. So it lets some things in and prevents other things from coming out. Just to review, let me show you back here. If we go back to this, this picture right here, with the cell wall is the peptidoglycan. So the cell wall is going to give the cell structure and strength. So it might prevent it from popping, but the cell wall is kind of hard and gives it structure. The membrane and the cell and any molecules can go in and out of the cell wall. Doesn't matter. It's like a basket. The membrane is going to be selective. Only some things can get through the membrane, and that's going to make the inside of the cell look different than the outside. So when we look at this gram-negative wall, we've got this outer membrane that's going to be selective. Only some things can get through. The peptidoglycan, being the cell part of the rigid part of the cell wall, is going to firm up that wall and make it strong. Okay. So cell walls give it structure. Cell membranes control what goes in and out of the cell. So I think of the cell wall kind of like a basket, and the cell membrane kind of much more flexible and fluid. And I'm going to stop right there.